So good morning, everybody. My name is Julio Maragall. I had the privilege to work in the University of Florida in the computational microscopy imaging lab. And today I'm going to present the pipeline that we worked in with codecs and H&E images. And this is for analysis and visualization of cellular data. I'm going to give you a quick outline. I will mention a little bit of my background, the skills that I learned with this pipeline, the problem that we're talking about, and how our objective as a lab and as a pipeline tries to tackle it. I'm going to give you a high-level introduction so I can go deeper in my methods and discuss and conclude about my results. This is where I come from. I am from Venezuela. I think that I embraced the more vibrant aspects of the country growing up, and I went to the United States as a teenager. I started my academic exploration in Santa Fe College, and I had the privilege to earn a fellowship to be a research assistant in the University of Florida. And I think that my scientific skill has become so much better. This has been a unique internship experience in the sense that the HuBMAP internship is normally extramural, and I have been working in this lab environment for 6 months now. So I got the opportunity to do HuBMAP research with people that I already know and have a degree of trust. So I think that my output was significantly boosted. These are the skills that I learned this summer. I won't list them off or read them off to you because I think that when I explain my pipeline, which was integrated into Python, and we want to publish it publish in the lab's HuBMAP, I hope you can grasp the skills that I had to do. And I am more than grateful that they gave me an opportunity to work with codecs imaging data, because there's prospective machine learning research that I want to do with kidney transplant, and preparing for this is truly a unique and fascinating opportunity by the HuBMAP incentive. So let me present to you the problem. We have the kidney, key organ, key component of the body, key regulator of physiological homeostasis, and it's one of the most structurally complex organs in the body. So the physiology and pathophysiology has so much diversity and so much complexity in the functional relationship, and there's so much to grasp from a tissue image, but the current histopathological approaches are unable to grasp the full breadth of complexity of the cell state. So there's this urgent need for an advanced technique to map and visualize and analyze structure function relationships at a single cell level, which is what the HuBMAP incentive does. The objective of our project was to automate an open source computational pipeline that processes, analyzes, and visualizes cellular information in codecs and H&E images. And why is our objective important? Well, on a very high level, we are enumerating and understanding diverse cell types. We're contributing to the more holistic view of the cellular landscape through the kidney, to the human kidney atlas and HuBMAP, because we hope that this project is generalizable to other organs, and well, it enhances an understanding of, in this case, the kidney's function and pathology. These are the two images that I work with. We have the H&E image. Just know that it's for visualization, it's histological, and it's titled, the H stands for hematoxylin, which is a stain that clings to nuclei. Then we have the codecs. This was one for analysis. This is a substantially bigger data structure because it has up to 60 markers or channels, and our bigger data set had 44 channels, and each channel clinks to different objects based on their DNA. So each channel can be representative and label and crucial to the kidney tissue that we're looking at. And to resume the pipeline, we have segmentation. We segment the nuclei and we get a spatial coordinate of where these nuclei are, and then we register the two images. And we have the codecs on top of the H&E. We do analysis to codecs. That's why I mentioned codecs for analyzing, and then we map it back on the H&E. So H&E for visualizing. And the first part is segmenting, like I said, gathering the spatial coordinate of the nuclei. So for the H&E, we applied a Python color deconvolution of the hematoxylin, which clings to nuclei, remember? And then we do intensity thresholding. And you can see that it recognizes the nuclei when they are isolated, but when they're on top of each other, it looks more like blobs. And so we needed to pass through more processing, which is the watershed segmentation and edge detection algorithm. And then we got in the end a cleaner product. And I say cleaner because our analysis is less noisy, thus more precise with this mask that we managed to generate with code. And it's replicable. Then then comes our analysis of the codex, which is feature extraction, manifold learning, and then the unsupervised clustering. So to clarify, my input data is it's codex, channel, image features, right? We have this variety of intensity, spatial, and morphological features, which totaled 626 for our 44 channel image. And we extracted at each location of the object, which is the segmented nuclei. And you see that there's a lot of nuclei and a lot of features. So it's a big matrix. And we have to perform manifold learning and dimensionality reduction to express all of these features extracted in the different channels on 2D. And that's when we get unsupervised clustering. In this case, it was hierarchical clustering. And now we want to see the relationship and try to see if the cluster predicts cell types and if our model actually understands the different cell types and potentially their relationships. So these are our results. And as you can see on the image on the right, you have the diagonal, which you see the hot orange cluster going through the diagonal, and that looks beautiful, that looks nice. We said when we looked at it, and it seems that it recognizes what an arteriole is, but how can we validate this? So we go into Sam, one of my mentors. He mentioned that I could look at the object or at the objects in a cluster and see if they show high intensity at a specific channel, because like I mentioned, the codex channels that we're analyzing mark different objects. So if it has high intensity objects in the podocyte channel, then that means that cluster is attempting to mainly segment or label or understand what podocyte entails. And channel in cluster 7, for example, it counted 88% of the high intensity objects that it counted on channel 25 were on a specific cluster. So that's our first clue that our model is actually understanding. As you see, the HuBMAP on top, 7, on the top, and there's like a little protuberance on the left part of the top structure. That's what it considers to be mostly podocyte. But then there's also co-localization, because the markers are not mutually exclusive. For example, the 2 circles that you see on the bottom right, well, that's the glomerular space. And you see, for example, in looking at another channel, you're seeing a glomerular space. And well, it knows that there's more podocytes in the podocyte marker itself than in the endothelial marker. However, there's co-localization, so there's intensity or high intensity in both markers, and that creates, let's say, a little more of a complicated analysis for the model. And this can be embraced as that we managed to see how clusters are compared and putting together by a computer, but also it has the drawback of co-localization in the sense that intensity can be mixed up. The arterioles, as we saw, that diagonal on the hot orange, came out to be pretty well recognized. As you see, cluster 3 in the HuBMAP is decently separated and represents about 90% of the arterial marker. And then we took this project and our results one step further, and we started looking at intra-cluster heterogeneity to make this as concise as possible. Intra-cluster heterogeneity is what variation within that cluster entails, and this is fascinating me, because we found out that 7% of the feature that extracted were explaining about 50% of the variance within a cluster. And then we can look at those features that we extracted and see what are the main channels that appear in those features. So, for example, we can see the word podocyte there, but then we also see arteriole or thick ascending limb. And then we start asking questions: how do this relate to each other? And do this indicate that there can be a level of correlation or even some degree of causation? And that's the higher level and goal, which is to try to get new patient outcomes, better treatments, try to find different innovative interventions, because we can generate maybe a podocyte signature in codex imaging, and we can maybe tailor interventions if we start exploring intra-cell functionality. So a takeaway of my pipeline, we have cellular identification. That's what it does, that's what it maps, and it's good for diagnosis, because maybe by counting, the clinician can see the different counts of cell types, and this can give quick and more precise diagnosis, especially on huge tissue images. We can look at intracellular heterogeneity and explore the different relationships and functions, and it can be applied to other organs. So as long as if it's H&E and codex data, we can try it on the heart, we can try it on the lung, and that's where we put it in on the lab's GitHub in the next month. And well, to wrap it up, I want to tell you a little bit about who I want to be, especially after this learning experience, because I think it has strongly taken me to want to pursue a PhD. I want to hopefully become a PI and research quantitative health, try to specialize in deep learning or signal processing in biomedical research. And I want to contribute, optimize, hopefully innovative quantitative methods to, well, computational nephrology. So one day after all of this effort, I can accrue sufficient knowledge to teach a younger generation of inquisitive individuals. I wanted to thank the lab, Mouse, Nick, and Sam for being my mentors, for giving me the crucial insight, for giving me the specific functions, Dr. Sauter for handling the duration and giving me the hard task, and all the lab for being just so receptive and encouraging for my development as a scientist. So thank you all for your attention.